So there's a lot of activity and clinical application that have happened in the last few years around microarray analysis and genomic analysis. I thought it would be good to have a session that might review and clarify some of the issues in interpretation and methodology. So we have two of our own in the region who are going to do that because they're experts in that area. David Ledbetter doesn't need an introduction, but he's the past president of SERGG and is professor and the director of the Division of Genetics at Emory, and Marilyn Lee, who is the director of the Molecular and Cytogenetics Lab at Tulane. Both of these will tag team. They've both been awardees of the ACMG Foundation Luminex Award for safe and effective genetic testing. So I'm really looking forward to this. So thank you very much, Dave.
Thanks. And it's nice that both the Constitutional Cytogenetic Array Consortium and the Cancer Cytogenetic Array Consortium are being led from individuals and institutions in the Southeast region. So I'll leave the Cancer Consortium to Marilyn. So Krista Martin and I started several years ago to arrange an informal consortium of labs to share data so that we would learn more quickly using empiric methods what pathogenic versus benign copy number changes were. We have a system now for laboratories to sign up via a website for this International Standards for Cytogenomic Arrays, and this is a purposeful takeoff on ISCA, the Standard Cytogenetics Nomenclature, also a voluntary organization. So over 120 labs have signed up, and the only obligation for that is to indicate your willingness to share data for both research and clinical purposes in a public database at NIH. We now have a new grant with a goal of 200,000 array cases in the next 2 years. The overall mission is to help develop standards for clinical interpretation for genotype data going into the database, phenotype data going into the database. As Hans alluded to, we initially had a grant from the American College Foundation sponsored by Luminex that allowed us to hold 2 initial workshops to discuss what the problems were and what topics could be benefited by discussion of standards. From the first workshop, the priority of everybody was to set up a central database that would be freely available. We've now done that at NIH. From the beginning, it was always the goal to include all technology platforms and all vendors to allow every lab to submit their data. And then to address this problem of how do you develop evidence-based guidelines for optimal design and, most importantly, clinical interpretation of arrays. From that first workshop, we were sort of pleasantly surprised because there were a lot of clinicians who at that time had not had tremendous experience using cytogenetic arrays in a clinical setting. But we quickly reached a consensus after discussion of the published literature that, in fact, the much higher yield of cytogenetics arrays and the relatively modest number of uncertain results meant that it was not only ready for prime time, but should be the first line diagnostic test for this category of unexplained intellectual disabilities, birth defects, multiple congenital anomalies, autism, etc. David Miller from Boston Children's was nominated to take the lead in doing a review of the literature and making recommendations that appeared in the May issue of the American Journal of Human Genetics as a consensus statement. And the consensus means lots of clinicians, lots of cytogenetic lab directors, lots of molecular lab directors, genomics, genome scientists, and bioinformaticians to recommend that chromosomal microarray should replace karyotype. And part of this, too, was to mount the evidence together in one place to allow people to be able to go to your insurance company. The insurance companies have rightfully questioned whether they should pay for both a karyotype and an array. This says if you only get to do one and the insurance company will only pay for one, we should all be doing array. and then only in selected circumstances would a karyotype have any value if you have a normal array result. The second workshop, we talked a lot about getting more data from normal controls, and the research community is providing lots of high-resolution copy number variant data from normal control populations. The NCBI not only agreed to set up their database in dbGaP and a newer database called dbVar for variant. They also went through the NIH IRB and got approval for an opt-out mechanism for submitting genotype and phenotype data in a de-identified format. We, at that time, did in fact implement the central database at NIH, created a website, and started web forums for discussions. We elected a steering committee, including clinical geneticists, genome research people, bioinformatics people, and diagnostic lab experts. There is a website that you can get the URL here, or Google ISCA, but there are a lot of ISCAs, so you need to add cytogenetics or arrays or IMRI to get you to the site. And then last year, during the stimulus grant opportunities with NIH, Krista and I put together a so-called Grand Opportunity Grant that was funded last fall, that has allowed us to put together several working groups for standardizing genotype information, phenotype information. Our primary partners in terms of laboratories are GeneDx and the Mayo Clinic, but also a multi-center prenatal diagnostic study comparing array to karyotype. That's Columbia, Emory, Baylor, and Signature Genomics, and all that data will be included, and then mechanisms for all other interested labs to contribute data. Before it goes to dbGaP and dbVar and NCBI, the data has to be de-identified and put into a particular format. We've had a number of discussions in a recent third workshop in Bethesda, where many of the vendors of array platforms and software were there. And the vendors are very interested in partnering with us and with their customer laboratories to develop software so you do your analysis in your clinical lab, and then you push a button that says submit data to NCBI. And you do that once a day or once a week. So the goal is to make it really easy for the busy clinical labs to submit their data to NCBI. NCBI will do some standard automated data quality checks, but then there's a big task that we're trying to figure out how to do, and that's how to curate the data and determine how do we officially determine something is a pathogenic copy number change, a benign, and minimize the variance of uncertain clinical significance. We're hoping that this project will also be a model for how to start addressing whole genome sequencing and getting whole exome or whole genome sequencing data from clinical labs into a central database, again, to speed the process of reducing the number of variants of uncertain significance. And this has some similarities to the Region 4 project collecting data from rare metabolic diseases in order to share large community data sets in order to extract clinically significant interpretations as quickly as possible. It's a Google map of the participating labs. There is also available what we call a consensus or community design at the beginning. Lots of different labs developed their own custom array designs, but when we got on the phone and talked to a lot of our colleagues, it turned out there were far more similarities in design than there were differences. And it was remarkably easy to talk through those differences and arrive at a consensus. So we pooled all of our designs into an agreed-upon consensus that's available initially on an Agilent platform, 44K, 60K, 105K, 180K. And the principles of this are similar to essentially all array designs now. We have oligonucleotide probes or SNPs or V-chip coverage of every chromosome arm with somewhat higher resolution near telomeres and near centromeres, and in known microdeletion regions or known genes that are known to cause intellectual disability, autism, other clinically significant disorders. And so you can create higher resolution but still cover the entire chromosome arm. As I said, these adjuvant platforms are now available directly from Agilent or Oxford Gene Technologies or Bluenome, but Affymetrix, Illumina, and Nimblegen all have the design files for this community array and are using those to inform their design on Affy, Illumina, Nimblegen arrays. So I think we're reaching a convergence of all of the platforms into a design that serves the cytogenetics and genetics clinical community quite well. And Marilyn will talk more about cancer design, which will be a little different. Now, how do we interpret it? So all labs are using a set of rules similar to the way that we've interpreted karyotype abnormalities for a long time. As you consult the literature, you consult OMIM, you consult all the genomic databases to see whether something is already known and has a specific phenotype. We also now consult the databases of copy number variants in the normal population. That's the database of genomic variants in Toronto, and the new DBVAR at NCBI. And then if you can't find that it's already known to be clinically significant or known to be benign, then you have to come up with another strategy. And we look at the size of the deletion or duplication. We look at what genes are included in that deletion or duplication to see if there are any Mendelian dominant haploinsufficient genes that might allow you to make an interpretation. And then, like all good cytogeneticists, you look to see whether it's a de novo event or inherited from one of the parents. And if the child has the same deletion or duplication as a completely normal parent, that will tend to shift the balance, although not be an absolute. So we'd like to improve our accuracy of this. Over time, in these normal CNV databases, we will be able to map the regions of the genome that, when deleted, when duplicated, do not have a phenotype. And then from the patient database, you'll know which regions do have a phenotype. And this is very similar to old-fashioned deletion mapping. You collect a bunch of patients with similar phenotype and overlapping deletions and define the smallest region of overlap or critical region that's responsible for the phenotype. And that region should never show up in the normal population if penetrance is high, and that's the pathogenic region. But there are a couple of things that we still have very incomplete information in. We don't know what the mutation rate for copy number variants, and it's likely to be different in different parts of the genome, and I'm not going to go into that today. A lot of the people who are new to copy number variants are struggling with incomplete penetrance and variable expressivity, a topic that geneticists are quite uncomfortable with. Although I think there's some confusion with copy number variants. When are we talking about incomplete penetrance? When are we talking about variable expressivity? And it depends a lot on whether you're talking about a dichotomous trait or a continuous trait. And I'll give you a quick example. If we take intellectual ability in the form of IQ scores, we're all familiar with the normal curve with an average IQ of 100 and standard deviation of 15. In syndromes that we're familiar with intellectual disability like Down syndrome and Fragile X syndrome, the mean is shifted so far to the left that most patients in that population fall below the cutoff, arbitrarily set at 2 standard deviations below below the mean. Although there are a few individuals that we refer to as high-functioning Down syndrome, high-functioning Fragile X, that might have a formal IQ score of 70 or 75. But they don't have IQ scores of 130. Now, if you go to other well-known syndromes like deletion 22q11, which is a copy number variant deletion 22, there's good IQ data, and the mean is 70. So it's significantly shifted to the left, and the standard deviation is 13. So it's a good normal distribution, just with a different mean value. So some people would argue you can't call this pathogenic because some people with 22q deletion have a normal IQ score. And we know clinically that when you see a child with 22q, about 10% of the time are the parents who thinks they're normal and is in the normal population, has the same deletion. Although when we call that result to the clinician and say one of the parents has the same deletion, they can usually tell us which parent has the deletion. So again, the parents are not over here in the normal distribution. They just don't meet our arbitrary cutoff of 2 standard deviations or more below. So now you can imagine when we find a new CNV where we don't have extensive IQ data, and suppose that new CNV costs 1 standard deviation in IQ, so that the mean is 85. When we have enough data, we're going to say this CNV is clearly deleterious, but it may not be pathogenic if pathogenic is a dichotomous label. So now I want to show some pilot data that allows us to start sorting out pathogenic and benign in a very straightforward and statistical way, analyzing 16,000 cases from 7 labs in the consortium. Some quick summary: those that are considered pathogenic are about 70% consistent with other large studies. But a lot of people are surprised at, because they have this misconception that clinical arrays are finding a whole bunch of tiny little deletions and duplications that we struggle to figure out their clinical significance, and that's not true at all. Most of the things we find are pretty big and just at or just below karyotype resolution, right? So they're 3, 4, 5 megabases, lots of genes. So the majority of our abnormal results are straightforward, dead easy to interpret. In our experience, about 5% of the time we need the parents in order to make a final interpretation. And when we get the parents, we're left with 1 or 2% variance of uncertain clinical significance. So 1 or 2% where there's a gray zone situation at the end of the result. But we've gone from a 5% clinically significant yield by karyotype to a 20% clinically significant yield by array. So most clinicians with that set of numbers think that the benefit far out exceeds the ambiguity of the variance of unknown significance. This computer's changed all my colors, but from this data set we can now look at mechanisms. Katie just talked about non-allelic homologous recombination caused by segmental duplications in the genome. So these hotspot recurrent deletions and duplications are about 25% of the cases. Those are the easiest ones to then determine which ones are pathogenic, which ones are benign. Although when they're first described and people find it in a proband and then it's present in a parent, they're not quite certain. So a lot of these new microdeletions, microduplications, you'll see conflicting reports in the first couple of publications. But when you have 16,000 cases and you start looking at the numbers of these, some of them are quite frequent. It's interesting that champion is 22q11 deletion, so it continues to be the most common microdeletion in human, even though a lot of patients with 22q deletion don't get array because they have a heart defect, and so it does FISH prenatally or postnatally. So This is probably an underestimate for things like Prader-Willi syndrome, Williams syndrome. This is probably an underestimate because these are only the patients who weren't clinically suspected of having those. Interestingly, the new microdeletion 16p11.2 with a very high risk of autism is number 2 in frequency. So it's a very common microdeletion with a very high risk of autism associated with it. So now that we have data sets that are getting large, we can take a formal case control, just like in GWAS analysis, and use that. In order to address this problem, we have some copy number variants we find in the normal population, some that we're certain are pathogenic, and then we have a whole bunch of variants of uncertain clinical significance. And what we want to do is get enough data to move all of them out of the variants of unknown significance into the pathogenic and benign buckets. And you can do that when you have enough data to support one or the other. So by doing that, and in this example, we've now compared our 16,000 patients with developmental delay, intellectual disability, autism, to 10,000 controls. And for things like Prader-Willi-Angelman syndrome, you never see it in the controls. DeGeorge syndrome down here, you never see it. So if you calculate an odds ratio, like GWAS studies do, the odds ratio is infinity because there's no denominator. And the p-values are astronomically significant. For other CNVs, you do find them occasionally in the normal control population, but you find them hugely more frequently in the patient population. Here's the 16p11.2 autism, 67 cases in the patient population, 5 in the normal control population, because most of these normal controls are not screened and phenotyped and said normal. They're just an assumed normal group of patients. So now with this approach, you can find all of these recurrent microdeletions reach statistical significance, except for the HNPP deletion, which is a neurologic condition that you will find in the, quote, normal population. Duplications are a little bit more complicated. Fewer of those reach statistical significance because they are found in the normal control population more often. Although using an odds ratio and statistical analysis, you can show that 1q21, for example, duplication is significantly overrepresented in cases compared to controls. And so you can conclude that it is deleterious, but you will see it in the normal population. In some cases, this helps us change the classification from a variant of uncertain significance to a pathogenic copy number variant. In other cases, it goes the other way. It goes from uncertain to benign copy number variant because it's present in controls as often as in patients. So we can do this for recurrent deletions. What about non-recurrent or random, which actually is the majority? It's going to be much harder. We're going to need much larger data sets, and then we're going to have to look at the overlapping region, and smallest region of overlap for deletions, etc. So we plan to continue to expand this database, and this will allow us then to post on the UCSC Genome Browser and other places. These are known pathogenic deletion regions. These are known pathogenic duplications. These are known benign deletions. These are known benign duplications. And hopefully, again, working with vendors, develop clinician-friendly portals and tools so that a clinician and genetic counselor who has a case can refer to this large database after it's been curated and compare their patient's data to this curated data set. And I've already mentioned Krista, who's my colleague who started this consortium several years ago, and we continue to work together on this project. Some of the other major labs in the initial pilot data analysis, Jim Ostel, Deanna Church at NCBI. And this is now funded by a GO grant from the National Institutes of Child Health. So I think we're going to have Marilyn come up and talk now, and then take questions after both presentations.